Hello everyone, I am Manoranjan Barman and I welcome you to my YouTube channel. Today I am going to explain the detailed working principle of Covaxin, Covishield, Oxford Astrazeneca and Sputnik V, then working principle of Pfizer, BioNTech and Moderna. In my last video I have explained different types of vaccines with example. So now my question is, do you know the example of killed vaccines which prevent Covid-19? The killed vaccines which prevent Covid-19 is Covaxin, which is developed by Bharat Biotech in collaboration with ICMR in India. The vaccine is prepared by killing or inactivating the virus. What about the recombinant vector vaccines? The example of vector vaccines are Covishield, Oxford Astrazeneca and Sputnik V. Oxford Astrazeneca vaccine jointly developed by the Oxford Vaccine Group and pharmaceutical company Astrazeneca to target SARS CoV-2 virus. Serum Institute of India's Covid19 vaccine called Covishield is a version of the Oxford Astrazeneca vaccine that manufactures in India locally by the Serum Institute Pune. The Sputnik V vaccine is introduced by the Russian State Institute Gamaleya National Research Center. Now what about the mRNA vaccine? The examples of mRNA vaccines are Pfizer, BioNTech and Moderna. Pfizer BioNTech Covid-19 vaccine are developed by Pfizer, an American multinational pharmaceutical company in collaboration with BioNTech, which is a German biotechnology company. And the Moderna Covid-19 vaccine is developed by American biotech company Moderna. Out of these three vector vaccines, Covishield, AstraZeneca and Sputnik V, the working principle of Sputnik V is bit different, even though all are categories under the vector vaccine. Before discussing the working principle of vaccines, let's briefly describe what happens when a person is infected by SARS CoV-2 virus. This is a SARS CoV-2 virus which causes the disease Covid-19. The SARS CoV-2 virus has its spike protein. With the help of the spike protein, the virus can attach with the cell receptor and enter into the host cell. And the mRNA is the genetic material of the virus which has the information for all the virus particles including the spike protein. Once the virus enters into the human body through nose or mouth into the lungs, then the virus will try to enter into the host cell in the lung. The virus can only enter into the host cell if the spike protein of the virus binds with the specific receptor of the host cell membrane. The whole virus particle will not enter into the host cell. Instead, the genetic material of the virus will enter into the host cell, and the RNA of the virus has codes for all the viral proteins. And in order to translate all the protein codes, the RNA will move towards the host cell ribosome. The ribosome will read the viral RNA and read the codes, and it will produce all the virus proteins like the spike protein and other virus proteins. And it will build a complete virus within the host cell. The virus may be more in number. Then the virus will rupture the cell and come out of the cell, and it will infect the nearby cells. Like this, the virus will make millions of copies and infect host cells and cause the disease. Till this point, the immune system will not understand what is happening around. Then the immune system will start working. There are some specialized immune cells called antigen presenting cells. These cells will engulf the virus inside it by a process called endocytosis. There are different types of antigen presenting cells. The most common are macrophages, dendritic cells. After the endocytosis, the cell will release some chemicals and destroy the virus particles into pieces. Some of the virus particle will be associated with some proteins of the cell, and it will be expressed on the cell membrane to the other immune cells. Then other immune cells like helper T cells, B cells and cytotoxic cells will be attracted and some signaling will go on in between them, and eventually there will be production of antibodies. And some of the cells will become memory cells for future reference. Then the produced antibody will bind with the virus spike proteins, and it will block the spike protein to enter into the cell and destroy the virus particle. So this was just briefly about the disease process of SARS CoV-2 virus. It is very necessary to know before understanding the working principle of the vaccines. Now let's talk about the working principle of Covaxin. This is an inactivated vaccine or killed vaccine. The inactivated Covaxin is not a new idea. It has been around for decades. The whole virus is taken and applied some chemicals to inactivate. The chemical used here is beta-propiolactone. Beta-propiolactone is the chemical which inactivates the virus. This chemical will inactivate the genes so that it cannot replicate and form a new virus inside the host cell and cause disease. But their spike protein will remain intact. After that, the inactivated virus are mixed with some adjuvants. Adjuvants can stimulate immune system to boost its response to a vaccine. The Covaxin is injected intramuscularly at the upper arm of the individual. Once the inactivated vaccines will enter into the individual, some specialized immune cells called antigen presenting cell will engulf one virus by a process called endocytosis. The antigen presenting cell will release some chemicals and destroy the virus particle into pieces. And some piece of the virus particle will be associated with some protein called MHC2 protein, and it will express on its membrane. The full form of MHC is Major Histocompatibility Complex. There are different types of MHC, MHC1, MHC2, MHC3. MHC2 is mainly present in the antigen presenting cells like macrophages, dendritic cells, and MHC1 is present in all the nucleated cells. You need to remember this point. Once the fragment of the virus particle is expressed on the membrane of the antigen presenting cell in association with MHC2 molecule, then T helper cells will be attracted and it will come and bind with the virus particle by a receptor called TCR, T cell receptor. After that, there will be some interaction or signaling in between the antigen presenting cell and the T helper cell, and then the T helper cell will be activated. Once the T helper cell will be activated, then it will secrete some chemicals called cytokines. There are different types of cytokines released by the T helper cells are interleukin 2, interleukin 4, interleukin 5, interleukin 6. These all are known as the cytokines. After releasing of the cytokines, it will attract and activate B cells, that is B lymphocytes. Once the B cell is activated, there will be clonal proliferation and differentiation. Clonal proliferation means increasing the number of B cells which can produce a single clone of antibodies. The differentiation means the B cell will be differentiating into some other cells called plasma cells. Once the B cells and plasma cells are derived, it will produce a huge amount of antibodies. So now you have the antibodies against the SARS CoV-2 virus after the vaccination. What if a real virus enters into the immune system? Then your antibodies will bind with the spike protein and prevent entering into the host cell and destroy the virus. Some of the T helper cells and B cells will remain as memory cells for future reference. The virus infected cell will take up some virus particles and express on its membrane, and it will attract cytotoxic T cells. Once the cytotoxic T cells come and bind with this, then cytotoxic T cells releases some chemicals. The cytotoxic T cells will release some toxic chemicals which will destroy the virus infected cells. Next is the working principle of Covishield and Oxford Astrazeneca. The working principle of Covishield and Oxford Astrazeneca are different from the Covaxin, as it is a recombinant vector-based vaccines. In this vaccine, instead of killing the virus, we use a piece of DNA that contains the information for production of the spike proteins of SARS CoV-2 virus. First we have to take the SARS CoV-2 virus, and then we have to take the spike protein. The spike protein helps the virus to enter into the cell. Then we have to take the genetic code for the spike protein and prepare a mRNA from those code. The mRNA is converted into a DNA, and the DNA will carry the code for the spike protein. That means from the DNA we can produce the spike proteins. Now let's see some basics. This is DNA. If we are converting this DNA into mRNA, it is known as transcription. And RNA to DNA is known as reverse transcription. It is going reverse, that is why it is known as reverse transcription. The mRNA will have some codes to synthesize some proteins. So if the code can synthesize the protein, the process is known as the translation. That means if the codes are converted into the protein from the mRNA, it is known as translation. Now let's go back to the previous slide. As the name suggests, vector vaccine. Now we need a vector to carry the DNA. The vector here is a chimpanzee adenovirus. Now you may think, why we are taking the chimpanzee adenovirus instead of human adenovirus? Our immune system may sometimes expose to some of the human adenovirus and may have built some kind of immunity against the human adenovirus. So we are taking the chimpanzee adenovirus, which a human body or human immune system have never exposed to that. The adenovirus typically cause cold or flu-like symptoms in human or in chimpanzee also. This adenovirus will only act as vector for the DNA that codes for the spike protein. The vector adenovirus cannot replicate within the human because the genes responsible for the replications are removed and the DNA is inserted into the adenovirus vector. In this way, the vector vaccines are prepared. The Covishield and Oxford Astrazeneca vaccines can be injected intramuscularly at the upper arm. Once the vaccine enters into the human body, the vector will carry the DNA and it will try to reach the host cell and enter into the host cell, forming a bubble. Then the vector will be broken down and the DNA will be released into the nucleus. It will use the host cell enzymes and it will try to do transcription and form a RNA. Once the RNA is formed from the DNA, then the RNA will move from the nucleus to the cytoplasm. Because the RNA will have the codes to produce the spike protein. So in order to produce the proteins, the RNA has to move towards the ribosome. The ribosome will read the codes and translate the proteins and produce the viral spike proteins. Once the spike proteins are produced, some of the spike proteins may stick outside the host cell membrane, or some of the spike proteins can escape out of the host cell. Then some of the circulating immune cell will encounter the spike proteins. One specialized cell is known as antigen presenting cell, another is B cell. B cell will simply interact with the spike protein and the antigen presenting cell will engulf the spike protein into the cell by a process called endocytosis. After taking it into the host cell, the antigen presenting cell will release some chemicals and it will destroy the spike protein into different pieces. The sum of the viral broken pieces will be associated with the MHC class II molecule and expressed on its membrane. Once the antigen presenting cell will express the fragmented spike protein, then T helper cell will be attracted and the T helper cell will bind with the antigen presenting cell with the help of T cell receptor and MHC2 receptor. And there will be some chemical signaling in between the antigen presenting cell and T helper cell, and the T helper cell will be activated. Once the T helper cell will be activated, it will release some chemicals called cytokines. These are interleukin 2, interleukin 4, interleukin 5, interleukin 6. After release of these cytokines, some other immune cells will be attracted, especially the B lymphocytes and the cytotoxic T cells. Once the B cell will be activated, there will be proliferation and differentiation of B cell and form into different cells like plasma cells and various clones of B cells, and eventually it produces antibodies. Once the immune system will encounter a real virus, then this antibody will go and bind with this spike protein and it will block the entry into the host cell and destroy the viruses. Some of the B cells and some of the T cells will remain as memory cells for future reference. And those virus infected cells will present some of the virus protein along with the MHC molecule and attract the cytotoxic T cell. Once the cytotoxic T cell will reach and bind with this receptor, then it will release some toxic chemicals and it will kill the virus infected cells. There is a little difference in the working principle of the Covishield, Oxford Astrazeneca, which the Sputnik V vaccine. For both the doses, in case of Covishield and Astrazeneca, they use the same chimpanzee adenovirus as a vector. But in the case of Sputnik V, they use different vectors for different doses. First dose they use adenovirus, the code is AD26. The second dose they used another adenovirus, that is the AD5. So they use different vectors for first dose and second dose. That is the difference. If you have the same vector for both the doses, then after the first dose, your immune system will produce antibodies against the spike protein, as well as your immune system might produce some antibodies against the adenovirus which your immune system has encountered. But in the case of second dose also, if the same vector is introduced, your immune system will have some antibodies already against that particular vector. So those antibodies might damage the vector before reaching the DNA into the host cell. So that is why if the same adenovirus vectors are used in both the doses, then the duration has to be long from the first dose and second dose, so that the antibody titer will be reduced till the second dose. That is an important factor if you are using the same adenovirus vector for both the doses. So as I have told you, in case of Sputnik V, the different adenovirus vectors is used for both the doses. So this is the major difference of Sputnik V with the Covishield and Astrazeneca. Now let's talk about the working principle of Pfizer, BioNTech and Moderna. It uses an mRNA which codes for the spike protein of SARS CoV-2 virus. The mRNA is not from the SARS CoV-2 virus, but it is synthesized. Let's see how this mRNA is synthesized. We'll take a SARS CoV-2 virus and take the spike protein. Then we will take the genetic code for the spike protein and synthesize an mRNA with these codes. Now this mRNA codes for the spike protein. Now we need to introduce this mRNA into the host cell as a vaccine. But we cannot do that because mRNA is very fragile. It may be damaged before entering into the host cell. So the mRNA is covered with a double layer of lipid nanoparticles and injected as vaccines. These vaccines are injected into the upper arm of the individual. Once the mRNA reaches the host cell, the lipid nanoparticles will fuse with the host cell membrane and the mRNA will enter into the host cell. This mRNA has the codes for spike protein of the SARS CoV-2 virus. So in order to produce those spike proteins, the virus has to reach the ribosome. The virus will not go to the nucleus. Once the mRNA reaches the ribosome will read the codes in the mRNA and produce the proteins. In this case, the virus spike proteins are the codes in the mRNA. So the virus spike proteins will be produced. And after production of these spike proteins, some of the spike proteins will come out, some of the spike protein will stick to the membrane, and the circulating immune cell will encounter these spike proteins as foreign particles. B cell will interact with the spike protein, antigen presenting cell will come and eat up some spike protein. It will break down and produce some fragments. Some fragments will be presented along with the MHC class 2. After presentation, the T helper cell will come and attract and bind with the T cell receptor, and there will be some signaling will go on in between the two cells, and the T cell will become activated. Activated T cell will release cytokines, the interleukin 2, 4, 5 and 6. So it will further attract and activate B cells. After the B cell activation, there will be clonal proliferation of B cell and differentiation of B cell. Clonal proliferation of B cell will produce numbers of B cells. Once the B cell will differentiate, then it will form plasma cell. Eventually it will produce antibodies. Now your immune system has the antibody against the SARS CoV-2 virus. When a real SARS CoV-2 virus enters into the immune system, your antibodies will bind with the spike protein and blockage the entry into the host cell and destroy the virus. Some of the B cells and some of the T cells will become as memory cells for the future reference. The virus infected cells, on the other hand, they will take some virus particles and express along with the MHC class 1 molecule and attract the cytotoxic T cells. Once the virus particle is expressed in association with MHC class 1 molecule, then the cytotoxic T cells are attracted and there will be interaction in between these two cells with the T cell receptor and MHC class 1 receptor. Then the cytotoxic T cell will release some toxic chemicals. These toxic chemicals will eventually destroy the virus infected cell. The cytotoxic T cells will not destroy the healthy cells. I hope you are clear about the topic. This is all for today. Please like, share and comment. And those who are new to my channel, please subscribe to my channel. Thank you.